Hello, my name is Shada Abachi, and I'm a PhD student at UC Irvine. And today I'll be presenting my talk titled Toward Evidence of Tune Inhibition as the Underlying Mechanism of Perceptual Metacognition via fMRI. When making a perceptual judgment about something in the environment, your confidence judgment or your metacognitive report usually tracks your accuracy. So when you are correct, you should have high confidence. When you are incorrect, you should have low confidence in your decision. MetaD prime measures your accuracy of this. However, there are situations where your confidence judgment incorrectly tracks accuracy. So when you are correct, you report low confidence. When you are incorrect, you report high. When do we see the incorrect tracking of accuracy? Well, under noisy and uncertain conditions, in mental disorders that affect metacognition, and in the lab where evidence of stimuli are unnaturally manipulated. A study done by the associate project scientist in our lab, Brian Maniscalco, is an example of where evidence was unnaturally manipulated. Participants were presented with a task where there was a noise patch and a noise and signal patch on either side of the screen, and an S1 type signal patch had constant contrast, while S2 had varying contrast levels. This made it so that the location of the signal patch is differentially predictive of how much information is present in that stimulus, and as difference in contrast between patches increases, participants should get better at the task. So as D prime goes up, meta D prime should increase as well. Overall, the black line shows that confidence accurately tracked accuracy. However, when participants responded S1, so the red line, as they got better and better at the task, their confidence no longer tracked their accuracy. Why might this occur? It's been shown that confidence tracks the accuracy of decision congruent evidence, which is known as confirmation bias in confidence. So is this idea of confirmation bias and confidence biologically plausible? Brian Maniscalco built a model of what we propose is happening at the neural level, where instantaneous evidence drives accumulators of absolute evidence, which then go on to excite units that have similar tuning preferences, but these units compute relative evidence, thus inhibiting those with opposite tuning preferences. These units that read out accumulated evidence of the preferred stimulus should contribute to confidence, and thus should have low levels of what we call inhibition tuning, since they are not inhibited by neurons of opposing tuning preferences. While these neurons should contribute to decision as they calculate the relative evidence of opposing stimulus preferences and have high levels of inhibition tuning. Based on our model, a decision is made when the activity of one of the differencing units hits a bound here, and confidence is read out after that decision, corresponding to the decision that was made. This is the figure we saw from the previous slide, and this is the results from the implementation of our model. The solid lines indicate the model simulations, while the dotted lines are the actual data. You can see that as D prime increases, meta D prime essentially breaks just for S1 responses. Great, so our biologically plausible model of tune inhibition matches actual human behavior. But is this what the brain is actually doing? In order to determine if that's what's actually occurring, we need to find the green decision and the yellow confidence neurons within the brain. And to identify these neurons, we use random dot kinematograms whose overall dot density, direction, and conflict, in other words, the balance of opposing stimulus choices, positive evidence, and negative evidence, are manipulated. And the combination of stimulus manipulations should allow us to identify which neurons are less inhibited, or the confidence neurons, based on how they should be preferentially activated to the combination of positive and negative evidence. We did this non-invasively in humans using fMRI, which gives us the advantage of whole brain coverage and human behavior. They were asked to report which way the overall direction of dot density was moving, left or right, as well as their confidence. To identify confidence and decision voxels in humans, we first simulated voxels given our model assumptions. Neurons with similar levels of inhibition tuning have been shown to hang out near each other within the cortex, and so voxels were either made up of either mostly green neurons or mostly yellow, as well as some percentage of inhibitory interneurons. And for our simulations and for the sake of our eyes, green dominant voxels are red and yellow dominant voxels are blue. And so in response to the different combinations of dot manipulations, we can see that plotting voxels' responses to conflict against each other shows that their distance from the identity line for both density conditions is related to the makeup of the voxels. We use this observation as a basis for calculating inhibition tuning levels. Inhibition index, or I index, can be defined as the activity in response to low conflict minus high conflict divided by low conflict. This means that voxels change their activity a lot when conflict increases, and those are the ones that are highly inhibited. So the inhibition index is bigger, and the ones that don't change their activity very much have a lower inhibition index because their numerator is about 0. And given our model, I index should be proportional to the inhibition level for both density conditions, and we also included voxels with different percentages of inhibitory interneurons to ensure that the relationship would still stand, even if an actual voxel had these different levels of inhibitory interneurons. We then use the observations from our simulations as motivation for the analysis of our actual data. To identify voxels of different levels of inhibition tuning, we were interested in decision-making areas such as the parietal lobe, as well as the most motion-sensitive region, the medial temporal lobe. And this is an example of the mask we use. The white is the anatomical mask of the parietal and medial temporal regions, while the red is voxels within those regions that significantly responded to density, which we decided based on our simulations. Within the voxels in red, we are interested to know whether I-index predicts confidence in our task. This is a cartoon diagram of what we predict our data to look like, where voxels with low inhibition levels should be predictive of confidence based on area under the curve classification, and voxels with high inhibition indexes should predict confidence at chance. This should give the relationship between the two a negative slope, and our paradigm should also not allow I-index to have predictive power of decision, so the slope there should be 0. For our actual data, we calculated the I-index of voxels and focused on those with I-indexes above 0, since those below 0 are probably doing things we aren't interested in for the sake of the study. Shown here are data from 4 subjects. We log transformed their I-indexes and plotted their AUC scores against them. The red lines are the slopes, and we found that the mean slope from all subjects of I-index versus confidence was significantly negative, while the mean slope for I-index versus decision was not significantly different from 0. This means that the data are behaving as our model suggests they should, and inhibition index can significantly predict confidence in precisely the way our model dictates. To conclude, the important points discussed were that dissociations between perceptual confidence and correctness of decision can occur, since decisions are read out by calculating the relative evidence of opposing stimulus preferences, while confidence is thought to track the magnitude of decision congruent evidence. Our biologically plausible model of tune inhibition was able to produce data that matched actual data of meta D prime breaking as participants were doing better at the task when they responded S1, and that there is evidence of tune inhibition within decision-making areas of humans using fMRI. Voxels with low TI were more predictive of confidence, and this predictiveness decreased towards chance as TI levels increased, since confidence should be read out by voxels of low inhibition, aka the yellow voxels, and that there was no significant relationship between decision and inhibition level. That inhibition level was not predictive of decision. Our next steps are to collect more data and ensure that these findings persist. And I'd like to thank Brian, Dr. Peters, and the rest of the lab for their support and their guidance, as well as UCI and the Air Force Office of Scientific Research for funding.